The primary research paper we chose was the convergent evolution of chicken Z and human X chromosomes by expansion and gene acquisition. The study of different chromosomes and their evolution has been actively researched for over 100 years. Chromosomes are structures that carry the genetic information of organisms that get passed down to their offspring. Sex chromosome evolution specifically has been highlighted and explored in many different species. In fruit flies, for example, it was discovered that the Y chromosome had degenerated and undergone changes, while the X chromosome had been preserved and unchanged. Along with this discovery, another scientist applied the same theory to mammals and birds, stating that while the W and Y chromosomes deteriorated, the X and Z chromosomes remained intact. Following this, the evolutionary relationship between the X chromosome in mammals and the Z chromosome in birds has been an enigma. The biological question that was raised in this paper was relating to whether the mammalian X chromosome is orthologous to the avian Z chromosome, meaning that these genes in these two distinct species originally came from a common ancestor and later evolved into the separate chromosomes we know them as today. The scientists hypothesized that the chromosomes shared a common origin. They were curious about the evolutionary trajectories between mammals and birds and set out to compare and contrast the gene densities and the sequences themselves.
Okay, for methods. The publication is focused on understanding the evolutionary similarities between the Z chromosomes in chickens and the X chromosome in humans. So, sequencing DNA. Scientists look at the genetic material, DNA, of the chickens and humans to understand the instructions in their chromosome. So, this is like reading an entire script of a play to see which character, in this case the gene, is doing. Comparing chromosomes. Compare the structure of the Z chromosomes in chicken and the X chromosome in humans. You can think this of like comparing two blueprints of different buildings to see how similar they are. Identifying genes. Identifying the specific genes in both chicken and humans to see which one is shared or unique. This is like finding a specific character in a script of a play to see if they appear in both stories. Analyzing genetic changes. Study how genes of the Z chromosome in the chickens and the X chromosome in humans have changed over time. This is like tracking two characters in a story and see how the roles have evolved through a plot. Okay, measuring DNA expansions. Scientists measure how much genetic material has expanded or grown in certain ranges of the chromosomes. This is like measuring how much the story has expanded by counting the number of pages that was added to it. Estimating DNA acquisition. Researchers estimate how many new genes in each chromosome has acquired. It's similar to counting new characters that are introduced in a new story. Comparative analysis. Scientists compare the chromosomes of chickens and humans with the aim to understand their similarities and their difference. This is like comparing two books and see how similar their storylines are. And sequences of technology. Scientists use advanced technology to read the genetic code more efficiently. This is like using a faster and more accurate method to describe create a script of a play. These experiments were like reading a genetic script of chickens and humans, comparing the stories, in this case the chromosomes, to see how they have evolved, measuring expansions, identifying new characters, in this case the genes, were introduced over time. The goal was to understand how the genetic makeup of chickens and humans have evolved in a way that is similar or convergent.
The methods described above provided the researchers with the following results. They were able to find that the chicken Z chromosome and the human X chromosome do not share a common origin. They evolved independently from different parts of the ancestral vertebrate genome. Both the Z and X chromosomes have undergone significant structural changes during their evolution, such as becoming less gene dense than their autosome partners and also becoming enriched for repetitive sequences. Both these chromosomes have also gained many genes, mostly testes expressed, to help in the development and sex differentiation. This suggests that convergent specialization for male reproduction, despite their heterogametic systems, exists in both birds and mammals. The following table from the paper, as you can see here, is what really shows the comparison of the structural features of the Z orthologous and X orthologous regions compared to the autosomes for both species. As you can see, in the Z orthologous regions of human chromosomes and human autosomes, the genes per megabase is lower in the Z regions. Same for chicken autosomes and the X orthologous regions. There seems to be a lower number of genes in the X orthologous regions or the sex coding regions. Now, we can see that there seems to be a decreased number of genes per megabase, which is followed by both of these in the same pattern. And it shows how both of these orthologous regions have lost chromosomes as they've matured and developed. This brings about a few implications that the scientists mentioned in their paper, and it was the fact that the independent origin, convergent evolution, the gene gain and gene loss of these sex-determining chromosomes challenges our traditional view of the passive ZX versus the degenerating YW. And it really shows how sex-determining genes, such as the human Y chromosome and the bird Z and X chromosome, have changed as they've developed over time. And we agree with the researchers and how they say that it opens new avenues for studying sex chromosome evolution and its link to male reproduction, as this change in genes and chromosomes greatly affects how we've studied sex chromosomes in the past and how we will in the future.
When making a conclusion in a scientific paper, you want to reference back to the hypothesis. In this research paper, it was sex chromosomes, chicken Z, and human X came from the same origin. To make a conclusion, we want to see if the results either support the hypothesis or reject the hypothesis, making it null. Based on the results, the hypothesis is null. Even though the hypothesis was not supported, this research is still important in terms of understanding the theory of evolution, the idea that all species have evolved over many generations. This research gives us more information on how living species are related and how they are created. With this in mind, we can explore future ideas. In the research, gene expression was found in the testes. One example of a future idea is to compare different genes beyond humans and chickens. This could look like using other animals that use one pair of chromosomes to differ the sex, or even use animals that use more than one chromosome pair. Using this model, it could be used to create a map of how different animals are related and the reproduction of animals in relation to evolution. And this is our works cited page. Thank you for watching.